So it's an honor. I'm very happy to be here with you today at the ICCB Latin America 2022. So my name is Anna, and I am a current PhD student at McGill University. I am in the lab of Dr. Livia Garcia, and we focus on the study of metastases in pediatric cancer. And my focus is on a typical and one of the most common pediatric brain tumors. So the topic will be elucidating the transcriptional landscape of metastatic medulloblastoma in patient-derived xenografts. Um, but first, a little bit of context. So we know that medulloblastoma is one of the most common pediatric brain tumors. It usually arises in the cerebellum, and it's so common that it's almost 20% of all the cases of solid tumors in the brain in infancy. And as one of the along the years, there has been like several studies on sequencing, and we have identified at least four molecular subgroups because medulloblastoma is, in the context of molecular heterogeneity, is super complex. And the most interesting thing about these molecular subgroups is that each of them will determine like the outcome of the patients. And even when we have identified these main subgroups, more robust sequencing technologies analysis have identified that within these four subgroups, we can have. So far, they have a total of 12 subtypes have been identified. And as I said, each of them will have an implication or affiliation to the malignancy and outcome for the patients. So for these molecular subgroups, one of them, the most aggressive is group 3. So I'm going to mention a little bit about this. So when we are talking about metastatic medulloblastoma, we are talking about something that we denominate as leptomeningeal disease, because it's the spread of metastatic cancer cells along the leptomeningeal space. And so it's fatal, and there is incurable, and no progress has been made in the 30 years to understand or to try to prevent leptomeningeal disease. So as I said, group 3 is one of the most aggressive subtypes of medulloblastoma. It has a great inclination or tendency to develop metastasis at some point, and 50% of the patients develop LMD at the moment of diagnosis. And as I said, the survival rate is very low, less than 5 years, and it's not been a real substantial improvement in the treatment. So we need to better understand how metastasis is developed in medulloblastoma. So as I said, it's no progress. And one of the most interesting thing is that metastasis in general is understood, and more in this context in the leptomeningeal disease, because it's very difficult to find samples. So and the reason is because patients that are diagnosed with LMD, with the abbreviation of leptomeningeal disease, there is no reason to operate, and it's very difficult to get samples post-mortem. So that's why it's so complicated to get samples for metastasis, and to develop a good study between the primary and metastatic compartments. But we need to find better strategies to identify which are the mechanisms that develop metastasis. And in that context, we can identify or hypothesis better therapeutic targets to prevent LMD. So what we are working here is in the lab is that we need to understand that, or the urgency for us is like we need to identify what are the molecular events that drive the subclones from the primary tumor to develop metastasis. So for this, we need to form models that resemble medulloblastoma primary and metastatic, that faithfully recapitulate the nature behavior of these two compartments. So once we have these models, we have aimed to perform what molecular profiling, and for example, I'm going to focus on the whole transcriptome of these two compartments to see if we can find genetic or molecular pathways that are enriched in the metastatic, and with that identify which are able for further validation in the context of functional genomics to see if we can extrapolate at some point to the clinic. So to establish the models of medulloblastoma, we have two approaches. First, we need to develop the primary medulloblastoma models that are representative of the human tumors. So for this, we have worked in collaboration with the University of Washington in Seattle, that they provide cell lines of patient-derived xenografts, which is just cells of tumors taken from the patients and injected into the place on site of the tumor in mouse. And then we passage this and we perform molecular analysis of these tumors coming from the mouse. So far they have been, all these patient-derived xenografts have been established, and we have different patient-derived xenografts for the different molecular subtypes of medulloblastoma, as I mentioned before. So this is a picture of our tumor here, primary. So when we identify the tumor, we do dissociation and molecular profiling. So far we have established these highlighted PDXs of medulloblastoma. So we are interested in group 3 mainly because this is the most metastatic. So once we have this, we do sequencing and we are ready for the bioinformatic analysis. For the metastatic model is a little bit more complicated because for that we need to we know that very rare cells from the primary left the central nervous system, travel to the through an hematogenous route, and then interestingly they go back to the central nervous system to invade and form leptomeningeal disease or metastasis. So knowing this concept, to do the metastatic model, we inject these cell lines in the flank. So we resect the primary tumor, and then we wait until we until we observe neurological symptoms that the cell have traveled back to the nervous system. And here, for example, we can see that the metastasis is at the base of the brain. So that's and it makes sense because as it is coming from the hematogenous route, we can see here all the spread of the metastasis. So we conduct the same analysis, we extract this metastasis sequence and perform bioinformatic analysis. And so just to validate our models that we were working with this with this accuracy of these PDXs, we do variant calling from RNA-seq data. So that's the pipeline to do. One of the main feature is that we are working with because we have the challenge that we can have contamination on mouse reads, right? So we do bioinformatic treatment to remove all the contamination from mouse, and then we can retrieve only the human reads. So we were positive, and we found mutations that resemble the nature of these patient-derived xenografts. So with that, we performed to do transcriptional analysis. So as I said, for us, it's very important to limit.
Hi, Anna. Sorry, you've accidentally muted yourself. If you could unmute yourself, thank you.
Sorry, do you hear me again?
There you go. Thank you.
Sorry, I don't know what happened. Share screen. Okay. Share screen. Do you hear me and see my slides?
We can't hear you yet. You can share your screen again.
You don't see my slides?
Not right now, no.
Yeah. Okay. Do you hear me and see my slides?
You're good, thank you.
Perfect. Thank you so much. Thank you for letting me know. So going back, okay, so we do differential expression analysis based on this pipeline that we can see here. And then the results are quite interesting. So what we identify is that indeed primary and metastatic are transcriptionally different. So we can see here in the PCA that primary and metastatic cluster separately from one another. So here I'm plotting three models of PDXs of group 3 medulloblastoma, and you see that there is a clear separation between the primary and mets. And as you can see here, for example, in the also in the PCA now by individual model and the heat map. And so what also did is like we did like enrichment plot to see which are like the pathways that are enriched in the metastatic, and we saw many pathways. For example, oxidative phosphorylation and protein secretion are one of the most that are taking our attention the most. And also because we have just a paper came out about that medulloblastoma uses GABA. In this case, the paper shows that metastatic development induces the oxphos metabolic phenotype. So it was good for us to see, like, we see a trend of these pathways that are enriched in the metastatic phenotype. But the most interesting thing is that we also conducted which genes in common were differential express and found among these three PDXs. And we only found, so far we have identified just one gene differentially expressed in all the metastatic PDXs of group 3 medulloblastoma, and this gene is FCGBP. And so we did we do some staining, and we see that in the signs of the tumor, we see a staining of FCGBP in the blood vessels next to the areas of the tumor, which is also very interesting because that makes it like to understand a little bit what FCGBP could be doing to enrich the metastatic phenotype. And also we can see that it has also been identified in another type of cancer, for example, in glioma and ovarian cancer. So it's not the first time that we see this gene enriched in other type of cancers. And also, like, in a collaboration with the University of Toronto, we were able to retrieve human data, not from PDXs but from human data, and we see that also FCGBP it's a little bit elevated in the metastatic compartment. And one of the first studies that were conducted to understand primary and metastatic, when we have data from microarray, we also see an elevation of FCGBP. And most recently, single cell RNA-seq data has been published, and we analyze like what is like the context, like for example of primary breast and lung cancer that have metastasized to the leptomeningeals, and we see that populations of cancer cells in the metastatic space has a FCGPP also highly expressed. So far this gene is one of the most interesting genes, and we are looking forward to understand what are the mechanisms for FCGBP to increase this metastatic phenotype. And just as future perspective, what we aim to do is like we have more genes, so we are working to understand what other genes are in common, and to see if we can pull more genes apart from FCGBP. But as I said, FCGBP is right now our potential candidate, and to do functional genomics in vivo. So far, right now, I'm working in this CRISPR system to overexpress FCGBP in a medulloblastoma cell line that we know that has a poor rate of metastasis. So we should see, if we overexpress FCGBP, we should see an increase of FCGBP an increase of metastasis. And Also, we aim to do further studies to pull the next of the genes that are in line to understand also which other genes and pathways could be enriching the metastatic phenotype. But as I said, here's, as you can see here, this is a combination of many things: bioinformatics, functional genomics, to try to understand like a very complex phenomena like such as the metastasis in brain. So this is my lab. Yeah, thank you for all the bioinformatics experts that always help me to get rid of all the problems that we have every day. Also, the trainees and postdocs in the wet lab and my facilities have always been so helpful. And thank you for your attention. I'm very happy to take any questions.
All right.